Hi, my name is Mansi Mehta, and I'm currently a third year PhD student at the George Washington University doing my research at the Center for Genetic Medicine at Children's National Health System. The lab I conduct my research in focuses on the human cytomegalovirus, or HCMV. Cyto meaning cell and mega meaning big is where this virus gets its name because cells that get infected with this virus tend to round up and swell. Hcmv falls under a specific class of herpes viruses, but not so similar to the aggressive herpes virus that most people immediately think of when they hear the term. It, however, does lead to adverse side effects in newborns, infants, and individuals who have a deficient immune system, such as HIV or AIDS patients. HCMV can also lead to babies being born with disproportionately smaller heads, and the infection can also cause deafness, blindness, and brain damage, very similar to that of the Zika virus. However, when healthy individuals are infected with this virus, they show almost no symptoms, and a large percentage of populations in developed countries are already infected with the virus unknowingly. There is currently no cure for HCMV, although many attempts have been made, and current antiviral drugs target portions of the virus that either mutate and evolve fairly quickly or have many adverse side effects themselves. Therefore, there is a dire need to further study this virus and how it continues its infectious cycle so that new drug targets can be studied, identified, and utilized for novel therapies. This virus works by first infecting one cell in the body and then hijacks the cellular machinery to produce multiple new viruses that break free from the infected cells and can go on and infect other cells in the body. In order to ensure its successful replication, this virus needs to prevent the infected cells from being detected by the immune system or dying prematurely before the new viruses are made. The process by which a cell undergoes programmed death is referred to as apoptosis. I aim to look at one important player in the HCMV replication cycle. Vimea is a protein that is encoded by the viral DNA, and Vimea, again encoded by the virus, has a main function, which is to prevent the infected cell from programmed cell death, or apoptosis, until the virus completes its replication, so that the immune system does not get rid of the infected cell before the virus is able to produce new viable viruses. Previous work has shown that without the Vimea DNA code, the protein does not get made, and the virus is significantly reduced in its ability to grow and replicate. Vimea also localizes to a specific compartment inside the cell called the mitochondria, more commonly known as the powerhouse of the cell, where it clusters and carries out its function by binding to and sequestering certain factors in the cell that promote cell death. Many other proteins in the cell also localize to mitochondria. However, what's intriguing and interesting about Vimea is that it gets made in an adjacent compartment to the mitochondria called the endoplasmic reticulum, or ER, whereas most proteins that migrate to mitochondria are generally not made in any cellular compartments and travel to mitochondria from the free space in the cell, or the cytosol. Studying this unconventional point of synthesis of Vimea and how it reaches its final destination at the mitochondria could provide valuable insights into how this protein is able to prevent the host cell from undergoing apoptosis long enough so that the virus is able to successfully replicate and continue to infect subsequent cells. It can also enable us to look at vemia as a potential target for drugs that can hinder its synthesis, localization, or functions and reduce the infectivity of Hcmv.